Hello, my name is Jonah Treitler, and the title of my talk today is Nutrient Profiles of Food Items in Adolescent Diet, a cluster analysis of data collected from a high school citizen science project. So first, let's start with some background. Many prior studies have examined adolescent diet and related health issues. However, the accuracy of dietary self-report data being used in these studies remains a challenge. For example, energy underreporting is well known, and the data collected on energy in some similar studies is found to be inconsistent with life, meaning that the data cannot possibly be accurate. So, multiple dietary assessment methods exist, each with their respective strengths and weaknesses. So let's look at some dietary assessment methods and what's good and what's bad about each one. So to start, we have the duplicate diet approach, which is good because there's a measurement of dietary exposures possible, for example, environmental contaminants. However, this method is not suitable for large-scale studies. The food consumption record is good because there's ease of application among those with low literacy or those who prepare most of their meals at home. However, individual dietary consumption is not accurate, and it is not suitable among those who frequently eat outside of their home. The 24 dietary recall provides detailed intake data with a relatively small respondent burden, as literacy is not required. However, there is possible recall bias. A trained interviewer is required, meaning there is possible interviewer bias. It's expensive and time-consuming, and it takes multiple days required to assess the usual intake. The dietary record provides detailed intake data with no interviewer required and no recall bias. However, there is a relatively large respondent burden, with literacy and high motivation required and possible underreporting. It is expensive and time-consuming, and multiple days are required to assess usual intake. Dietary history assesses the usual dietary intake. However, it is high cost and time-consuming and not suitable for epidemiological studies. The food frequency questionnaire assesses usual dietary intake simply. It's cost-effective and time-saving and suitable for epidemiological studies. However, it is specific to study groups and research aims and uses a closed-ended questionnaire and has low accuracy with recall bias. So, in a novel citizen science project, 28 students in the T.C. Williams High School in Alexandria, now renamed Alexandria City High School, successfully collected detailed lifestyle data each day from December 14th to January 14th. Dietary data here was collected using dietary record, as the citizen scientists found that this would be the most appropriate method for their purposes. And because the students are motivated as citizen scientists and students in a science class, the data collected by the project is potentially more accurate and complete. Furthermore, the inclusion of both school and non-school days also made the data more representative. Now, why citizen science, you may ask? The citizen science approach, which refers to the scientific projects involving citizens' participation, can benefit study subjects and researchers. The students serve as both the study subject and the researcher, both the data source and the data analyst. Several studies have demonstrated the benefits citizen science projects bring to the scientific community and the development of scientific knowledge. For example, AI for Mars and the Christmas bird count. So let's talk about the purpose of this project. So our goal was to characterize the adolescent dietary patterns using dietary records from a citizen science project. However, the main problem that we fall into is that many food items do not fall neatly into a single food group, like fruits, vegetables, grains, etc. So our solution for this is to cluster the food items according to their macro and micronutrients, with macronutrients being nutrients such as carbohydrates, proteins, fiber, cholesterol, and fats, and micronutrients such as calcium, potassium, sodium, iron, zinc, and vitamins. So for data preparation, we took 3 steps. The first step was processing, where we extracted individual foods, corrected typos, and identified any distinct foods, because naturally in the data there were repeat foods, as people often ate the same things. And so for some of our purposes for data analysis, we needed to organize a distinct list of these foods. The second step was nutrient lookup, where we used USDA as the primary data source and 100 grams as the standard serving size. Finally, we did a data quality check, where we checked any outliers, fixed any missing values, and corrected any duplicate records. For all of the food items in the data set, which is 3,948 total food items with 674 distinct items, the minimum, mean, maximum, and standard deviation was calculated and reported by nutrient. Using the FDA's definition, the percentage of food items with high and low nutrient was also reported by nutrient. So, the FDA's definition of high and low nutrients is guided by the daily value percent, where 5% or less of a daily value is low and 20% or more of the daily value percentage is considered high. And the percentage daily value tells you how much a nutrient in a serving of food contributes to a daily diet. And this diet is based off of the 2,000 calorie diet and is used for general nutrition advice. So a cluster analysis was performed using R. The analysis used 2 different clustering methods, k-means and Gaussian mixture modeling, also known as GMM. The cluster mean and median were identified and normalized using the recommended daily values for each nutrient to form the cluster profiles. So let's look at k-means. Well, here k responds to the number of clusters, and the means refers to the average of the data in a cluster, that is, the centroid. The algorithm assigns a data point to the nearest centroid, and it also tries to minimize the within cluster differences between data points. As you can see on the figure on the right, you can see how k-means works. GMM is a probabilistic model for representing normally distributed subpopulations. It uses a technique called expectation maximization, or EM, to find the right model parameters. However, the number of clusters, like k-means, does need to be predefined. So, to determine the number of clusters, we will use the Bayesian Information Criterion, or BIC. This number that we get from the BIC will be confirmed using the ELBA method. As you can see on the figures on the right, using the maximum value of the BIC, we can associate that with the number of components, or the clusters, which in this case is 9. And this number is confirmed by looking at the ELBA method, where the curve in the data happens at about 9 clusters. So, protein, sodium, and energy have the highest medians, and all of these medians are normalized using the aforementioned daily values. And potassium, cholesterol, and calcium have the lowest medians. Now, however, most of the foods are not high in any of these nutrients per serving. The nutrients of these 3,948 food items were retrieved and they were analyzed, and as you can see, 31% of the food items were high in protein and 24% of the food items were high in sodium. However, most of these food items were not high in anything, but rather they were low. So as you can see, calcium, 76% of these food items were low in calcium, and 92% of all of these food items were low in potassium. So let's look at the nutrient profiles. 9 clusters were generated by k-means and Gaussian finite mixture modeling using the expectation maximization algorithm. The largest clusters showed similar nutrient profiles from both methods. However, the smaller clusters were somewhat varied. There were more clusters with higher macronutrients than micronutrients, though the largest cluster is low on almost all nutrients except for sugar. This is confirmed by the figures on the right. So now for a discussion. We performed clustering analysis on a novel citizen science dataset, and to the best of our knowledge, the clustering-based nutrient profiles are a new way to characterize adolescent dietary patterns. We found that the nutrient profiles suggest that the adolescent diet lacks more micronutrients than macronutrients, and the largest cluster of food items lacks in both except for sugar. However, there were some limitations to our data. For one, there is no data on the number of servings, as our citizen scientists found it very hard to determine the number of servings exactly. And secondly, there was a relatively large number of food items from a small number of individuals. So in the future, we would like to analyze the differences between school and non-school days, and we'd also like to validate the profiles using additional data. So here for acknowledgements. We'd like to thank the teachers and students at Alexandria City High School for their roles in data collection. We'd especially like to thank Ms. Jennifer Uche, a former Alexandria City High School science teacher. We'd like to thank Dr. Linda Zanin and Ms. Terry Capshaw for coordinating the GWU Biomedical Center's collaboration with ACPS and FCPS under the Go Virginia Region 7 grant. And finally, we'd like to thank GWU and TJHSST, that is Thomas Jefferson High School for Science and Technology, for their support and commitment to the development of our next generation of STEM experts. Thank you for showing interest in my topic.